Hi folks, this is the second part of chapter 9, and this is going to start on page 320, module 9-9.4 in your textbook. So if you have that with you, go ahead and open that up. One thing that we want to remind ourselves of is this picture right here, which is from the previous video. And we want to kind of remind ourselves of all these lines, bands, and zones, and especially the purple, bluish here, thick filaments, and these reddish, pinkish, thin filaments. So we're going to talk about what is a thick and thin filament. What are they made of? The thin filaments are attached to the Z lines. So if we look right here at the Z lines, it actually looks like a Z, with actinin. And they're composed of 4 main proteins. So if you look over here, the F-actin here, if you look at this bracket here, is going to be the double strand of these red spheres, which is the G-actin. And then you can see the tropomyosin, which are these cords, these rope-like cords, and the troponin, which is sitting right here. The F-actin is F for filamentous. It's a twisted strand of the G, which globular, if you really want to remember those two. The G-actin is the important one because it has an active site or parking spot for binding to the myosin heads, which is part of the thick filament. Nebulin is going to hold the 2 G-actin strands together. Tropomyosin is a twisted protein, and you can see it kind of looks like a rope here. Whereas the troponin is attached to the tropomyosin and it makes this complex. It also attaches to the G-actin, these red spheres, and has a binding site for calcium. That's going to come into play in our next videos. The thick filaments are myosin molecules. And if you can kind of picture this myosin molecule here and how many of them are twisted together, okay, that is why it looks thick. It's because of these myosin molecules twisted together. Then there is titin. This connects the thick filaments to the Z lines. The myosin molecule is itself, if we look at one individual, it's composed of 2 twisted subunits, and then it has this free head part that works on a hinge, and you can kind of see here, this is going to move, and that's going to be key in the sliding filament theory. This head of the golf club is going to move. And then the tails are twisted this way. There's a free head, and this head that hinges is going to form a cross bridge with actin. So that's really key. This part of the free head of the myosin molecule is going to form a cross bridge with the subunit, the actin subunit, with the parking spot, with the active site. And so then it's going to act like a hinge, and it's going to pivot. This is going to pivot, and it's going to move things closer together, and then it's going to let it go. So here you can see this one right here is good, and this is something we're going to talk about soon, hopefully, sarcomeres. But this one subunit here, we're going to observe the sliding filament theory. So this head is going to attach to these actin subunits, these G-actin subunits at the active site, and then ATP is going to make it hinge, and when it hinge, it's going to shorten this subunit. And that whole idea is the sliding filament theory. So when muscles contract, thin filaments slide over thick filaments due to that cross-bridge binding and the hinge. So that means that, if you remember from the first slide here, the H and the I bands get smaller. So here's an I band, and here is an H band, so they're going to contract, it's smaller. The zones of overlap are actually going to get larger. So this right here, where there is overlap, is going to extend because the I band here is shortening, and so this is what you get. I'm thinking we probably already talked about sarcomeres, but if not, it's Z line to Z line here. This is this unit. Sliding occurs in all sarcomeres and each myofibril. So individually, you have it shortening due to the hinge and the myosin and the actin, okay? And then as the myofibrils shorten, so that's all sarcomeres in each myofibril, and then as myofibrils shorten, the muscle contracts. So individually this is happening, but when you have multiple units, then you have the entire muscle contracting. This is a video that's going to show you kind of what's happening here, and it's also going to show you the ATP involvement and this calcium, and this calcium is going to come up in a later video. All right, now we're going to get to how these sliding filaments kind of get the message to start contracting. Okay, so let's go ahead and jump to page 322. We're into the next module here. And we want to talk about, what kind of triggers all this. And this is very chemical in nature. So, dust off those chemistry lessons in your brain. And biology, really, because remember we talked about transport across the membrane. This is going to review that as well. So all cells in the body are polarized, meaning there's positive and negative charges that are unequally distributed on both sides of the plasma membrane. Typically, the inside of the cell is a little bit more negative compared to the outside of the cell. So I think we did this in one of the previous chapters, the extracellular fluid and intracellular fluid. That's what we're going to be talking about here. The fact that there's unequal distribution, that there's a concentration gradient of the ions, and there's a membrane involved, that is referred to as membrane potential. It's measured in millivolts, and the resting membrane potential is -70 millivolts. So the skeletal, and that is just something that you should just put into your brain, because when we talk about neurons later in this chapter and then in the next chapters, that -70 is kind of like a baseline. Skeletal muscle fibers, they have a resting potential of about -85 millivolts. So let's kind of visualize that, and that we're going to look at what's on page 322. So if we're measuring it, we would get -70 millivolts normally, okay? Slightly more negative on the inside than on the outside, okay? Extracellular fluid, intracellular fluid, that's going to be abbreviated here, ECF and ICF. But you can see there's a difference, okay? Now, the plasma membrane we already talked about being selectively permeable. The cytosol and ECF have different compositions. So there's more sodium outside in the extracellular fluid, and there's more potassium inside in the cytosol. Negatively speaking, most proteins are on the inside and they're too big to cross. So there's more chloride ions in the extracellular fluid, but not much of it diffuses across. There are leak channels, which are proteins embedded in the plasma membrane, which allow a slow flow of sodium and potassium, okay? And this is a natural diffusion down their concentration gradients. So there needs to be pumps to help export sodiums out and import potassiums in. So this uneven distribution with the leak channels, but the ion pumps kind of overcoming the leak channels, that is what maintains the resting membrane potential. So we're going to talk about something called an action potential. And the picture on page 323 is going to be the best one to look at here. Okay, and this is going to be repeated. Some of this information is going to be repeated when we talk about neuromuscular stuff and the nervous sections. So there's 5 steps, and if you kind of follow along with the chart on the top of page 323, this will make sense. So step number 1, and actually I might want to do that too. So step number 1, there's a small increase in the sodium ion permeability. So what happens? Sodium is entering the cell and it changes the membrane potential, okay? It changes it and it moves it towards its threshold of negative 55 millivolts. So negative 55 is the threshold for an action potential to occur. Okay, so that's what you see here is it's moving up towards this negative 55, okay? At that threshold, step 2, voltage-gated sodium channels open and positive charged sodium ions rush in. So that's depolarization. You get this huge rush of positive sodium ions. And that helps to change the membrane potential to positive and reach this plus 30 millivolts. So here we are, all these sodium is rushing in until we hit this plus 30 millivolts, okay? This plus 30 millivolts, and when we hit that, the voltage-gated sodium channels, they close, and then potassium channels open, okay? And this is called repolarization. So that means that sodium is leaving the cell, causing this repolarization, and the membrane potential to come back down. I'm sorry, I'm skipping around. This is how I like to see visuals when I'm explaining. So we're coming back down. And then step 4, repolarization continues, and some of those potassium channels start to close. And then at 5, the membrane potential stabilizes, okay? And that's when the potassium channels are closed and we're restoring the original distribution. We're going back to that negative 70 millivolts. And the refractory period is the time needed to originate the distribution. So the time needed before we can get another stimulus, open up the gates, and start another action potential. So here, that is right here. And you can see the refractory period right here until we get back to that negative 70. Okay, so from that negative 55 to back to that negative 70. The propagation of an action potential is through neurons. Okay, both neurons and skeletal muscle fibers have these membranes that are electrically excitable. Okay, and the depolarization, repolarization, so that curve that we saw, it produces this action potential that spreads along the plasma membrane of both neurons and muscles. And when we talk about the neuromuscular junction, you'll kind of see how this spreads. It's going to travel only in one direction, and it allows kind of a very quick communication, right? So stay tuned for the neuromuscular junction, which we are talking about in the next video. Here is a video on action potential. Truly, this is probably better for the nervous system, but you get the idea and you can see kind of what's happening along this curve. And this is my friend Bozeman, not really my friend, but one of my favorite videos to watch. So bring your questions to Friday discussions, Friday clinical days, and or email me.